So I'm a third-year medic. My name is Rohit, and I'm leading this lecture on essentially inflammation and everything that is encompassed within it. So in this lecture, we're going to talk about cell death and repair. We're going to talk about what inflammation actually means, how we can prevent it, or we can reduce it. And then we'll talk about specific immune response, which is essentially how we deal with pathogens when they enter our body and infiltrate our body. So oh wow, okay, my notes come on as well. So if we could join this Socrative room, it would be great. So essentially you would just have to go onto this website called socrative.com and press onto Socrative, log in, and then choose student login, and then press on the write the ID. So okay, so don't answer the question yet. We will get onto the question eventually. So yeah, so leave the question be for now, but we will get onto it. Okay, as you guys join, I will move on. I will remind you guys again anyway, so it shouldn't be a problem. So the whole point is that I don't want to talk the entire time, and it's nice to have some MCQs in between, so you can sort of get some understanding and idea of how the questions will be in your exams. So we're going to first talk about cell death. And in broad terms, cell death is broken down into two types of cell death. One is apoptosis, which is basically mediated by caspase enzymes. And these are, there's two types of caspase enzymes that you'll come across, or two broad classes of caspase enzymes. And then there is necrosis. Now, the difference between apoptosis and necrosis is that apoptosis is regulated, it's controlled, whereas necrosis isn't. And the downstream effects are very different. Now, apoptosis causes the formation of these things called apoptotic bodies. And they're essentially eaten by phagocytes or taken up by phagocytes and then destroyed. And so that promotes the resolution of inflammation. So then it would cause the macrophage to release IL-10 and other repair markers like TGF-beta, and that will lead to the repair of the tissue and stop inflammation. Whereas necrosis will promote inflammation because it produces DAMPs, which is damage-associated molecular patterns, which we'll get onto. And an example of that would be like ATP. Because ATP is inside the cytoplasm of cells, it would naturally be released in necrosis because the cell just releases all of its cytoplasm, and then that would then promote inflammation through IL-1, IL-6, IL-12. So there is a difference. Now, in terms of the enzymes you need to be aware of for apoptosis, there's two sets, which I have not written on here on the slide, but there's two sets. There's initiator caspases and executioner caspases. So initiator caspases are the caspases that get activated upon detection of damage. So they are your caspase 2, caspase 8, caspase 9, and caspase 10. And then there's executioner caspases, which are your caspase 3, 6, and 7. So when you get damage, caspase 2, 8, 9, 10 get activated, and then that leads to the activation of 3, 6, and 7, and that causes apoptosis. It's just a bit of an extra bit of knowledge that you need to know for the exams, because they do ask. So I thought it was worth mentioning. So as we said, one promotes inflammation, one doesn't. So we're going to talk now about inflammation because it's sort of on that topic. So first thing first is I've sort of done it in like an animation format because I think it's best to understand inflammation in almost like a story. So firstly, there is inflammation of a tissue caused by the damage to a tissue because of bacteria that have entered the area. Now, those bacteria can do a bunch of different things. One is that they can damage the tissue, which then leads to these DAMPs being produced. So DAMPs are anything, again, as I said, that signals damage of a tissue. And these include uric acid, so that would be in joints. ATP, that would be if a cell has necrosed because of damage. Or DNA, again, because a cell is necrosed, then it's released as DNA. But then there's also PAMPs, which are pathogen-associated patterns, and they essentially, examples are LPS and exotoxin. So LPS traditionally, if you remember from your lectures in first term, is activates your TLR4, which is a example of a PPR or PRR. So I will give, so essentially these receptors that bind to these dampened PAMPs are called TLRs, NLRs, whatever, but they are forming a broader class called PRRs, which are these receptors that are encoded within these cells like macrophages. And the first step of detection is always detection because of tissue resident macrophages. These include Kupffer cells, which are in the liver, microglia, which are in the CNS, or just tissue resident macrophages in tissue or muscle. So they will detect it. Now, once they detect it, it causes these macrophages to become pro-inflammatory because it's detected some form of inflammation. So when they become pro-inflammatory, it means they release things like TNF-alpha, IL-1. And for the exams, you need to know about these systemic effects. So TNF-alpha will act on inside of within your brain, but also on your hepatocytes, and it promotes this process called acute phase reaction, and that's basically where the liver tells your body we're going to upregulate some inflammatory proteins and we're going to decrease other proteins like albumin, because we want to push all the amino acids we can into the production of these inflammatory proteins. So these include things like complement proteins, so like your C3 alpha, C1q, whatever. And then we don't really need albumin during inflammation because we're trying to kill the bacteria before we restore normal homeostasis. So we sort of deal with that by acute phase reaction. And that's really important because we'll get onto why that's important in a second, actually. And then IL-1 is another systemic cytokine you need to be aware of, and they test you in the exam. Essentially, IL-1 is a thing called a pyrogen. A pyrogen is nothing but a cytokine that basically promotes increase in temperature of the body. So in this case, it's good to have a pyrogen because one of the key signs of acute inflammation is rubor or heat, and that's important because you want to promote increased metabolism of macrophages and whatnot in order to kill off the bacteria as quick as possible. There's also other cytokines that are released, or like NO and prostaglandins, and all of that contribute to increased migration of these cells in order to deal with these pathogens. Now, one of the things we were talking about was acute phase reactants, and one of them is called sialic acid. And for the exam, you guys need to know how basically these leukocytes, these white blood cells, migrate from inside of a postcapillary venule to the damaged tissue. And there's essentially a few steps to it. Now, the first step to it is margination, and that happens when all of these red blood cells, if I go back, all of these red blood cells initially weren't coagulated, they weren't coming together. But what suddenly happens is that because of this acute phase reactants, they start coalescing together to form this thing called a rouleau, and that basically pushes all of these white blood cells towards the sides of the venule. And that's important because as soon as they get pushed to the side, they then start rolling across the endothelium. And when they're rolling, there's an interaction between a bunch of receptors that are on the white blood cells and some essentially receptors that are on the endothelial cells. There's three, and they're called selectins. Now, there's three things you need to be aware of, three of those selectins. They're called P, E, and L selectins. And the word they are, it sort of makes sense. So P selectins are in platelets and endothelial cells. L selectins, L stands for leukocytes, so they're in immune cells. So the L selectins on the leukocytes will bind to the E selectins, and E selectins are E because they're found on endothelial cells. So the L selectins on the leukocytes will sort of very weakly bind with the P and E selectins that are found in the endothelial cells, and they would sort of just roll around, just rolling across the endothelium. Now, if we go back to when we said, there was a release of cytokines from the macrophage, That's what happens. Now, when that release of cytokines happens, it causes the activation of these endothelial cells, but also these immune cells, because these are small enough molecules that they can actually move through the endothelium. And so when these go and activate the endothelial cells and immune cells, what actually happens is it causes an increased transcription and increased expression of these other important binding proteins called ICAMs and integrins. Now, ICAMs are present on endothelial cells. Integrins are present on white blood cells. And this time, instead of it being a very weak essential attraction, weak binding, this time it's a very strong binding. So as soon as that happens, they stop moving, and now they perform diapedesis. Now, diapedesis is just a big word just to say they're moving from the blood vessel to the tissue. And the way they do it, you need to be aware of for the exam. And that's through these proteins on the white blood cells and on the endothelial cells called CD31. I haven't mentioned all of this, and they will be found on my PowerPoint, which I will distribute at the end if you guys fill out my my form, which I'll explain later on. But essentially, there's CD31 proteins on the white blood cell and the endothelial cells, and they will essentially bind together, and that will basically push the white blood cells through the endothelial cells in between them and allow them to then sort of move into the tissue. So now they've moved into the tissue. But if you remember from your first term lectures, not all the cells that are found in the tissue are the same as the cells that are found in their blood. So for example, you don't really tend to find many macrophages within the blood as you do comparatively to if you find them in tissues. Normally you find macrophages as monocytes. And so what happens is that these monocytes migrate into a tissue. So they diapedese, they perform diapedesis, and then they differentiate now. So these monocytes now essentially change their form and become a macrophage. And that's really important because monocytes aren't inflammatory intrinsically, whereas macrophages are. Now, neutrophils, they remain as they are. Neutrophils were always neutrophils, and they remain as neutrophils even after performing diapedesis. And we will talk about what these do. So monocytes not only also differentiate into macrophages, actually, they also differentiate into dendritic cells. That's the other one, which we will talk more about later on in the lecture when we talk about specific immune responses, because I think it gets important then. So now they're in the tissue. So now they actually need to destroy the pathogen. Now, the way they destroy it is three mechanisms. Now, the three mechanisms are broken down, or at least I think the best way to see it is phagocytosis, non-oxidative mechanisms, and oxidative mechanisms. Now, phagocytosis you might have been aware of from A levels, but essentially the bacteria or pathogen, whatever the pathogen is, it will be engulfed by a phagocyte. An example of a phagocyte is macrophages, includes dendritic cells, even B cells can be examples of phagocytes. And so they will engulf the pathogen and form a phagosome, and then that binds to a lysosome to form a phagolysosome, and then it essentially breaks it down. So it's quite important for phagocytosis, and that plays a big role in actually allowing for immunity to occur later when we talk about it. Then there's also non-oxidative mechanisms, and they're methods that don't actually involve oxygen intake. So they're mechanisms that don't require oxygen, and they can just be performed by a cell in the absence of oxygen. And they've been performed, experiments have shown this because they can put it in a non-oxygen environment or deprivate it with oxygen, and it can continue to produce these things. And these include hydrolytic enzymes. So one of the examples you'll come across is things like perforin. There's things like AMPs, which stand for antimicrobial peptides. An example you may have come across is LL37. You may have come across it if you did some of the lab stuff, which I'm not surprised if you didn't. And then there's also cytokines and complement proteins, which we'll talk about later. But then there's also oxidative mechanisms. So these require oxygen, and experiments have shown that there's an idea called respiratory burst, and that's how this has come about. That they found that when you put it in oxygen, these cells suddenly show a sudden increase in oxygen uptake, and the reason for that is because they need to produce things like nitric oxide, so NO, or reactive oxygen species, or hydrogen peroxide. The important enzymes you need to be aware of for these is NO is made by an enzyme called INOS, which stands for inducible nitric oxide synthase. And that basically converts L-arginine, so an amino acid, into nitric oxide plus citrulline. So essentially, the arginine gives an electron away and a nitrogen atom away to oxygen. And in doing so, it makes nitric oxide, but because it's lost an electron in a nitrogen, it's converted into a citrulline. And that's how it works. And that's nitric oxide. Now, nitric oxide can just oxidize bacteria or viruses, whatnot. So it's very good at doing that. Then there's also reactive oxygen species, and that's formed by NADPH oxidase. And one clinical link you may want to write down is a link with a disease called chronic granulomatous disease, and that's basically where there is a mutation in the NADPH oxidase. And what that means is that the reactive oxygen species can't actually be made. And so if they can't be made, then you're more susceptible to infection with bacteria that are very dependent on oxygen. So yeah, that's essentially what ROS does. And then H2O2, which is hydrogen peroxide, can actually go on to make hydroxyl radicals. So it produces H2O2, and then that reacts with oxygen and water within the tissue to make hydroxyl radicals that together will oxidize the bacteria and the pathogen. So yeah, so that's essentially what the idea was, that there's three mechanisms. One uses oxygen, one doesn't. One basically engulfs a pathogen, and in doing so, it basically kills off whatever pathogen would have been in the tissue. And yeah, and that's what happens. Now, for the purpose of the exam, you also need to be aware of the complement system and kinin and fibrinolytic cascades. Now, I've left out the formation of the fibrin because that was a first year content stuff. But essentially, if you remember, the idea is that fibrin is made because fibrin is converted from fibrinogen to fibrin, and yeah, and then it can be broken down through a fibrinolytic cascade, where basically you activate an enzyme called plasmin, and then the plasmin will basically break the fibrin down into smaller monomers, breaking down the clot. Now, in terms of these classical pathways, there's three mechanisms. If we look on the left-hand side of the diagram, basically there's three mechanisms by which you activate the complement cascade. So this is the entire complement cascade. The core of it is always the same. So anything below C3 is the same. So C3 goes on to activate C3, becomes C3b, and then the C3b activates C5 convertase, and then that makes C5a or C5b, and then that can do either inflammation or make a macrophage membrane attack complex. So the important ones you need to be aware of, which I've sort of written in the PowerPoint, is the C3b is for opsonization. So if you don't remember what opsonization means, it's essentially a way to tag pathogens or cells that have been infected with either viruses or bacteria to be phagocytosed. And so it's like a good marker for it. Then there's C5a and C3a. Now, they're what is called anaphylotoxins. It's just a big word to say that they help to degranulate molecules, help to degranulate immune cells, and degranulate just means to expel all of their granules of enzymes or of histamine or whatnot. Now, then the C5b to C9. So essentially what happens is that all of these C5b will then bind to C6, C7, C8, C9 to form this thing called a membrane attack complex, or a MAC complex. And that basically is a massive pore in the cell. So it can basically drill a hole within certain bacteria to basically cause osmolysis. And the caveat is is that it only kills gram-negative bacteria. So it's not gram-positive also. So, and the reason for that is that gram-positive have very thick membranes, so they're not susceptible to MAC complexes, whereas gram-negative have very small, very thin membranes, so they are very susceptible to it. Now, the reason why I put the diagram on the top is that there's also, this is the kinin cascade, and basically the idea is pre-kallikrein becomes kallikrein, and then kallikrein makes bradykinin from basically hydrolytic enzymatic degradation of kininogens into kinins. But what kallikrein also does is that it also activates plasminogen and converts into plasmin. So kallikrein also interacts with the fibrinolytic cascade and activates that. And it's just a minor point on the spec, but I thought it was worth mentioning anyway because it's still fairly relevant, and it may be asked in the exam, which is why I mentioned it. But bradykinin, so kinins, an example of that is bradykinin, and bradykinin is also an inflammatory mediator. So what it does is it promotes massive inflammation by causing arterial vasodilation, by releasing prostacyclins or nitric oxide. So it promotes nitric oxide release from immune cells, but also from endothelial cells. And it can also make veins contract with other prostaglandins. So in all doing so, bradykinin helps to promote a very, essentially a very edema sort of inflammation, which is important because it wants to push as much permeability through so that as many immune cells can come into the damaged tissue. Okay, so last thing we're going to talk about is essentially the difference between acute and chronic inflammation. And we're going to essentially, there's 4 ways you can say the differences are. The first difference is timeline. So the first difference is timeline, and that is that essentially acute inflammation lasts for a few minutes to days, whilst chronic lasts for weeks to years. And the reason for that is more the causes of it. So it's a direct consequence of that. Now, whereas acute inflammation is caused by damage of tissue because of infections or trauma, so like say someone cuts themselves because they were cooking or something, then that would be a reason why acute inflammation would occur. And these are very easy to repair and to deal with because infections are very easy generally to deal with by the immune cells. But the difference is in chronic inflammation, the infections that they have are very much dependent on the inability to either complete the inflammation or to resolve the inflammation. So what that basically means is for example, TB. TB is a disease that tuberculosis is caused by a mycobacterium, and that basically will live within the macrophages in our lungs, and it can't be killed off until another group of cells interact. And I'll talk about that in the next slide anyway. So bear that in mind. Then there's also autoimmune cells, autoimmune diseases like MS or rheumatoid arthritis, where the length of it is caused because there's a constant antigen, which is our self-antigen being present, which constantly activates immune cells that are reactive to it. Then the third one is silicosis, and the fourth one is chronic osteomyelitis. And they're linked together because the idea is that silicosis is non-pathogenic, and like it itself is an antigen, but it doesn't actually cause any pathologies. It doesn't cause any damage. But what it means is that the cells are constantly being affected by it, and it causes granulomas to form, which we'll talk about. And then chronic osteomyelitis, again, is a chronic inflammation because it can't repair the tissue because it's in the bone, and it's really hard because there's so little vasculature within the bone comparatively to the rest of the tissues, it becomes really difficult to repair. And so what ends up happening is there ends up forming a devitalized bone, so bone ends up dying. And then that leads to another chronic inflammation occurring, another bout of it occurring. Now, then the other two you can look at it is gross features and histology. And the way you can look at gross features for acute inflammation, which you need to be aware of, is, I think the easiest way is to look at through the Latin, so tumor, dolor, rubor, calor. And essentially the idea is that all of these mechanisms will help with migration of cells, with dealing with increased metabolism, but pain because of peripheral sensitization. So if you go back to neuroscience, some of the things like bradykinin and prostaglandins actually promote nerves to become more sensitized to pain. So they'll show a lower threshold to signal. Whereas chronic inflammation doesn't really show any pus because it tends not to be a pyogenic infection that causes it, so an infection that causes pus. There's very little edema, again, because not many cells are migrating at this point, because since it's been lasting for weeks and years, it's more mediated within the tissue now rather than external cells within the blood coming into the tissue. But the histology is very different between these two. Whereas acute inflammation is dependent on PMNs. So PMNs mean polymorphonuclear cells. Essentially, they're called polymorphonuclear cells because if I go back to this page, so you can see a polymorphonuclear cell within you can see two of them within the postcapillary venule. They're called that because they have three segments that look slightly different from each other. So they're called polymorphosis to multiple different looking nucleus cells. So an example of that is neutrophils. So essentially in acute inflammation, the major component is neutrophils, when there's some macrophages in it, and there's a lot of pus and there's very little fibrosis because it doesn't last for very long and there's no remodeling of tissue occurring. Whereas in chronic inflammation, there's literally no PMNs. It's mainly mononuclear cells like macrophages, lymphocytes, and then the macrophages can form other things like giant cells and epithelioid-like macrophage cells. And there's a lot of neovascularization, which means new blood vessels forming and collagen deposition. So because it's chronic, it means that there's always a bit of repair occurring of the tissue, but it can never completely just sit into that repair phase. And it just ends up doing a bit of inflammation and then repairing, a bit of inflammation and then repairing. So it ends up forming some collagen as well within the tissue. And you can see that in histology slides. Now, the last thing I'm going to talk about is granuloma within inflammation. So in simple terms, granuloma is just a structure that is formed within the lungs or within anywhere actually, where if the bacteria or the pathogen can't be destroyed, it's basically encased in a bunch of macrophage rings followed by a T lymphocyte cuff. And what you need to be aware of basically is that the reason why initially the macrophages can't kill the tuberculosis, the mycobacterium in this case, is that the mycobacterium, when it enters the phage lysosome that we were talking about before, when it does phagocytosis of the mycobacterium, it can't actually end up then breaking it down. So it ends up just sitting inside the macrophages, and it ends up forming these foamy macrophages, which is because they're just taking up all the mycobacterium. Now, as soon as T lymphocytes detect this, they start releasing something called IFN gamma, so interferon gamma, which is an interferon class 1. And essentially what that basically means is that these encourage cells to suddenly start showing viral or ability to deal with intracellular pathogens. And so suddenly this gives the ability for macrophages to start killing these mycobacterium, and that's through multiple mechanisms. One is oxidative mechanisms. And so then suddenly you start seeing these macrophages start dying off because they start killing off the mycobacterium within themselves, and in doing so they die as well. But then other macrophages start becoming more activated because suddenly there's more DAMPs and PAMPs inside the central core of it. And so then they release TNF alpha, causing more macrophages to aggregate. And that is why tuberculosis tends to be a chronic inflammation. And then the mycobacteriums that haven't been killed off then remain, so that afterwards they can then kill off other, they can then again activate macrophages to again do chronic inflammation. Okay, so we're going to do some Socrative questions now. So I've talked enough, I guess. So if you guys can go on and answer the first question, that would be amazing. Nice. So the majority of us answered. Yeah, so the answer to this should have been macrophages. And the reason for that is that neutrophils do produce TNF alpha, but macrophages are the main source of TNF alpha because they need it for their own accumulation and activation. Neutrophils do, they just don't produce as much as macrophages do. And yeah, and that's why in granulomas, macrophages are very important in order to form that granuloma cuff around the mycobacterium.